So chapter 5, it's about the integumentary system. So what we're going to learn today is the components of this system, and then we're going to describe the functions, and as well we are going to consider the subcutaneous tissue, which is not part of the integumentary system, but just because it's close by, we consider it in this chapter. And also we're going to see the specific details of the epidermis, the dermis, what types of skin do we have. We're going to see as well what are the factors that contribute to the skin color. And we're going to see, for instance, the classification of burns. And we're going to see the characteristics of the derivatives of the integumentary system, not only the skin, but also the hair and the nails, which are accessory structures. And we're going to see some examples of the exocrine glands, the growth of hair, and sebaceous and sweat glands as well. So, the integumentary system is the largest system in our body. So we have a skin, we have like around 1.5 to 2 square meters and makes about 4 to 5 kilograms. So each kilogram has 2.2 pounds. So let's say that someone has 5 kilograms of skin. So if you multiply 5.5 times 2.2, that, 5 times 2.2, that gives you more or less around 11 pounds of skin. And then attaching to the skin, we have the accessories, which are the hair and the nails. And the hair is not located in areas where we have thick skin, like the palms and the soles. Now, in the skin, we have 2 major layers. We have the epidermis and we have the dermis. And they are subdivided into several layers. We will see which ones they are. Functions of the skin: to protect against injury, to protect against infections, to prevent dehydration, and as well the production of vitamin D. And we help as well to regulate body temperature with the skin. In the skin, we have several receptors that helps as well to give a sensation. So we can tell if an object is hot, if it's cold, if a texture is smooth, if it's rough. And then we have receptors for pain, which are the nociceptors. So if something is activating these receptors, you have the sensation of pain. The skin is multi-layered, and it has cells that give rise to these different layers, but it also has specialized cells that give the skin color, and cells that binds foreign molecules that we call foreign antigens, and it protects us from these foreign antigens. So the accessory structures, which are the hair and the nails, provide a kind of complementary function because it also protects. So for instance, our nails help us to grab objects, but also, just in case, it helps for protecting not only the fingertips, but against aggression, for instance. And the hair, it helps to basically maintain our skin with the lack of, for instance, dust that may go directly into the skin if we didn't have the hair. So the hair basically protects the skin from excessive debris to coming into there. So, again, components of the integumentary system: skin. And the skin, if you remember, it's a membrane. It is the cutaneous membrane. Hair, nails, and accessory glands, which are sweat glands, produces sweat; sebaceous glands produces sebum, that oily secretion that maintains our skin lubricated. And then the subcutaneous layer, which is not part of the integumentary system, but it is important for cushioning. So this is located just under the dermis. So what is the function again? Protection, sensation, thermoregulation, water conservation, immune system, and synthesis of vitamin D. So now let's look at the epidermis. What layers makes the epidermis? And the dermis as well, and the subcutaneous layer. So the epidermis is made out of keratinized stratified squamous epithelium. So if you remember, epithelial cells surrounds our outside of our body and the inside of our body. So we have, in this case, several layers of cells. That's why it's stratified, and the cells are squamous. The cells are flat. And on top of these multiple layers of cells, we have a thick layer of keratin. This keratin, it's an inert protein, so basically it's non-living. And this one prevents us from dehydrating. It acts like a seal on top of all these layers of cells. Now the papillary dermis, which is just below the epidermis, is made out of loose connective tissue. So below the papillary dermis, we have the reticular dermis, which is made out of irregular connective tissue, dense, irregular. So we have collagen fibers there that are thicker than the ones that we have in the areolar layer, and these are close together. That's why it's called dense. And they randomly orient themselves within the skin, within the reticular dermis, forming like a net. Reticular means net. Rete, red in Spanish. And then underneath this reticular dermis, we have the subcutaneous layer, which is made out of loose connective tissue and adipose tissue. So the skin is made out of connective tissue and epithelial tissue. The epithelial tissue is a stratified squamous epithelium. That's the epidermis with keratin. Underneath, we have this areolar connective tissue, the loose connective tissue, and then we have the dense irregular connective tissue. So, because in the top layer, in the epidermis, we have an epithelium, this epithelium doesn't have blood supply. It's avascular. But the dermis is vascular. So we have a lot of blood vessels there. And these blood vessels provide the oxygen and nutrients and other molecules that this epidermis needs to be healthy. And then the subcutaneous tissue also has abundant blood supply. So here is a diagram showing you the integumentary system. So we have this top layer, which is the epidermis, and then we have this bottom layer, which is the dermis. All of these, epidermis and dermis, form the cutaneous membrane, and below we have the hypodermis. And you can see here in this hypodermis these blood vessels. The red ones represent arteries that brings oxygen-rich blood, and the veins represents deoxygenated blood, so blood that is rich in CO2 rather than oxygen. And another thing that you can see here in the dermis is that the dermis, it connects with these finger-like projections that are called papilla. Papilla means fingers. So you have here the papillary dermis, and then you have the dermis connecting like this. So these extensions of the epidermis are called retipex, and then they connect with the papillary dermis. Why do you think we have this type of connections? Why the epidermis binds like this to the dermis? Why not just flat like this? It's easier for the attachment to occur if you have this, because you have less surface area. And with this, you increase the surface area. So if you were about to put your hands like this, one on top of each other, and you will ask someone, try to separate my hands, and you do this. Do you think the person pulling your elbows to the sides can separate your hands easily? Versus doing this? Which one is easier to pull apart? This way or this way? The first one, right? This one is easier to pull apart than this. So it's like Velcro. So this is why we have this. And then here in the dermis, we have accessories like these glands, sweat glands, sebaceous glands here. This is the hair follicle. And then we have receptors. We will see each of these receptors, its function. So we have two categories of skin, thin and thick skin. They vary in number of layers. In the thin skin, we have 4 layers. In the thick skin, we have 5 layers. And we have accessories in the thin skin as the thick skin, but in the thick skin, we don't have hair or sebaceous glands, but we have a lot of sweat glands. Where do we have thick skin? In the palms and the soles, and thin skin everywhere else. Sometimes, depending on the activity of the person, that thick skin can become thicker. So you develop an extra, you don't develop an extra layer, but your keratin layer becomes thicker. Like if someone shovels a lot, you will start developing a lot of keratin in this area. So that's a callus. Okay, so here are the components of the epidermis. So we have a strata or layers. So this one, which is the first layer, it is called stratum basale. So this one is the first layer. So underneath here we have the dermis, and this is the epidermis. So the first layer, it is the stratum basale. So in this layer we have the stem cells that makes the these layers of cells. So these stem cells are keratinocytes that divides by mitosis. So in this mitosis, we have 4 stages of cell division. So the first stage, it is prophase. So I'm going to talk about the cell cycle right now. So we have something in histology that is called the cell cycle. Cell cycle has 2 major components. So if you make it to look like this, so you have a big part of the cell cycle that is called interphase. And in interphase, we have a segment of this that is called the G0 or gap 0. Sorry, G1. And then we have something that is called the S phase. So this one will be S phase. And then we have a G2 phase or gap 2. So between interphase we have a stage or a phase that is called the mitosis phase. So in mitosis, we have 4 parts. The first one that is called prophase. The second one that is called metaphase. Third, anaphase. Fourth, telophase. Most of our cells are not constantly dividing. So they are in interphase. So when they are about to divide, they go from the interphase, G1, they go into the S phase. So this is where you duplicate your DNA. So here what happens is you duplicate your DNA. And hopefully, during this S phase, you don't make any mistakes when you are duplicating the DNA. So we have enzymes, like they are called DNA polymerases, and they're supposed to add, like, according to the sequence that you have, like for instance, if you have adenine, thymine, guanine, thymine, adenine, guanine, cytosine. So they're supposed to make the complementary bases when they make a copy. So what it will be the complement for A? T. What about for T? A. And then C, A, T, C. So hopefully your DNA polymerase doesn't do this. Because in here, what it's supposed to be? G and G. So then you replicate or you duplicate your DNA during S phase. And then you make organelles, and then we have checkpoints here during G2 that if you have a mutation, and if that mutation is something that the body cannot, or the cell cannot tolerate, then that basically that cell is not going to live. Sometimes in certain situations, the cell salvages itself. It's like if you have a car and then you have a car accident. Sometimes insurance total it, so because there is no repair that can be done to the car. So that happens during these checkpoints. So now let's say that the cell is ready, it has normal DNA after S phase, it has everything ready in G2, it has the number of mitochondria that is needing, the number of lysosomes that is needing, everything is ready. So now it can go into all of these that we call mitosis. Now what happened during mitosis? Well, in prophase, let's say that this is a cell, and this is the nucleus. So this is the cytoplasm. In prophase, what you have is, you have in S phase all the chromatin, like the DNA, it's like all like this. So in prophase, what you do, it is you condense your chromosomes. And you start disappearing the nuclear membrane. So prophase, visible chromosomes, and disintegration of nuclear membrane. As well, your centrioles, let me put it like where it starts. Centrioles migrate to opposite ends. Of the cell. So you have two centrioles. So one of these is going to go into here. Okay, so that's prophase. Now, in metaphase, the next phase, so we already did this. In metaphase, chromosomes align to the equator of the cell. And microtubules attach the center of the chromosomes. Now, chromosomes when they condense the chromosomes, they look like this. So you have two sister chromatids. These are called sister chromatids. And then this is the center. So this is called kinetochore. And then you have two arms. Let me sharpen this. So this will be the arms. One is going to be longer than the other one. The long arm is going to be the Q arm. The short arm is the P arm. So then, these are the chromosomes that were condensed during prophase, and now look how they're going to align in the equator of the cell, which is the center. And then by this time, you don't have a nuclear membrane, but then you have your chromosomes aligned like this. So this is the equator, and no more nucleus, of course. So now this will be metaphase plate. At the cell's equator. And then the microtubules start forming to attach to the center of these chromosomes. So all of these together is called mitotic spindle. And then with this mitotic spindle, you will pull the chromosomes apart. Once you go into anaphase, the next phase. So we consider this. So any questions about prophase or anaphase? Okay, now let's move into anaphase. So in anaphase, you have shortening of these microtubules. So these are the microtubules of the mitotic spindle. And then these chromosomes, these sister chromatids that were together, they start separating and going in into different poles of the cell. So these are sister chromatids pulled apart. How far? Well, not so much. So anaphase chromosomes separate into separate or into individual sister chromatids. Due to shortening of the microtubules of the mitotic spindle. And not only that, so this would be sister chromatids. Not only that, but nuclear membrane starts to form again. So then you start making a new nuclear membrane around this. Is there a name for the little slices or like little segments of orange once you peel an orange? In Spanish we call them gajos. But I don't know in English. Anyone? Like you know that you peel an orange and then you can segment it, right, into tiny pieces.
Segments.
Segment is even for my body. So we don't have, for instance, agajos for the body. Like if someone get a burner and then they put it into parts, we don't call them, it's gajos de persona. We don't call that. So yeah, that's kind of one of those words. Anyway, so let's pretend that the nucleus was an orange, right?
It's called wedges.
Wedges, yes. But it's generic, but it's good, yeah. So how are you going to get the wedges out of an orange or a tangerine without basically taking apart the nuclear membrane? So that happens in prophase. You disappear this nuclear membrane, and then the microtubules attach to these chromosomes. And then once you split them apart, you form another nuclear membrane. So it's like, let's say you eat half of the orange, and then you want to keep it prevented from drying, but you were very careful when you're peeling the skin of the orange to keep it so that you can protect your wedges, the half. And you don't have Saharan wrap. So what would you do? You have your wedges there, your half of your wedges, and then you coat it with the skin. And then you eat it later on that day, and then it won't be super dry, those wedges. Does that make sense? So now you start putting back a nuclear membrane. You start forming again. Not all together, because you still need to pull them more apart, but that happens in telophase. So Any questions of anaphase? Now in telo means ending. In Greek theater, there is like these big curtains that they put at the end of the performance. They close it, that's telo, that's the end. So at the end of mitosis, you have telophase. So in telophase, definitely there is no more need to have these microtubules because you already shortened so much the microtubules that your sister chromatids are farther apart. So sister chromatids farther apart and wrapped inside a complete nuclear membrane. So you form again your nuclear membrane and you start as well pinching the cytoplasm so that you can have two different cells. This is happening during cytokinesis. Now something important. So this is the furrow, the cleavage furrow. So something important about mitosis is that you're supposed to have equal numbers of of chromosomes, an equal number of organelles. So you only achieve it then when you have the right steps going on. So you're supposed to have equal number of chromosomes per daughter cell. If you're going to have more than one child, make sure, guys, that you have equal amount to give to each one. I mean, not because the youngest is young, is not going to know. Once he gets old, he's going to also want same money as the eldest.
What happens in G0 phase?
Yes. So not all the cells undergo mitosis and continue the cell cycle. Some of them, they exit, they go into G0. In G0, basically the cell don't divide anymore. So do not divide. So these are terminal cells, like our brain cells, our muscle cells. You have a specific number of neurons, a specific number of lymphocytes, muscle cells, and if you damage them, you won't get anything else anymore. Any other questions? So then in this stratum basale, that's where you have the dividing keratinocytes. So you have stem cells. And these stem cells are constantly dividing, and replacing these superficial cells. And as they divide, the cells migrate from the stratum basale which is here, into the next layer, which is called the stratum spinosum. As the cells move from the stratum spinosum into the next layer, which is the stratum granulosum, look at what happens to the cell size. It decreases. Why? Because the cytoplasm starts becoming less abundant. And then you develop some granules of protein that is called keratohyalin. And then the organelles start dying. And then if it's in thick skin, you will have a next layer that is going to be very thin, almost translucent. So this is the stratum lucidum. So you don't have nucleus in this stratum lucidum, as you can see here. In all of these cells, you have a nucleus, but not in this layer. Now, the last layer is going to be the stratum corneum. So in this case, this is dead keratinocytes, so it's only keratin. So, stratum basale, stratum spinosum, stratum granulosum, stratum lucidum, and stratum corneum. A lot of layers. How can you remember them? Well, there's a mnemonic that if you go from superficial to deep, like the most superficial is corneum, is the one that basically your skin touches the air. So that's the most apical layer. So there is a mnemonic for that. So these are thick skin layers. So corneum, lucidum, granulosum, spinosum, and basale. We have the same in thin skin, except lucidum. We don't have lucidum. So to remember, California likes girls in string bikinis. So here in this stratum spinosum, we also have these cells that are called dendritic that allow us to trap foreign antigens and start an immune system reaction. And then here in this stratum basale, where we have these stem cells that makes these keratinocytes, we also have some receptors that are called the tactile cells that are connected into a nerve cell. So these help us to have sensations of soft touch. And then we also have specialized cells that produce a pigment that are called the melanocytes that gives our skin the color that we have. So here is a view of thin skin and thick skin in light microscopy. So if you see, this stratum corneum in the thick skin is very thick. And looking here, is very little layers. So that's something that you will look in the microscope when you look at the skin. Very thin stratum corneum in thin skin versus in thick skin. Now for the dermis. So the dermis has this papilla. It has this papillary plexus of capillaries. So these capillaries are the ones that gives the oxygen and nutrients that this epidermis needs. And then we have corpuscles that are found in between the papillary layers that are called tactile corpuscles. Similarly as to the tactile cells that we have in the stratum basale of the epidermis, these ones help us to detect light touch or smooth touch. And then at the bottom of this dermis, we have the lamellated corpuscles. These ones help us to detect true touch or deep touch, and as well vibrations. And we have these blood vessels from the subcutaneous tissue that helps to provide blood supply to this reticular layer. So this is how then these different layers look like in the dermis. So this is the papilla. It's part of this areolar connective tissue. So in here is where we have a lot of elastic fibers. And then here we have the dense irregular connective tissue with some blood vessels. And then we have in thick skin epidermal ridges that basically forms our fingerprints. So these are the epidermal ridges. And then opening of sweat pores in these epidermal ridges. Now in skin we have orientation of collagen fibers that are known as cleavage lines or Langerhans lines. These are part of the reticular dermis and are important for surgery. If you try to cut the skin following those lines, the skin will heal faster than if you cut across. So these cleavage lines then indicates the constant tension or the predominant tension of the muscles and the stretching of the skin. So if you see them, you can see the orientation regularly, especially the ones in the face. I mean, they're not really well shown in here, but they are important for performing perhaps cosmetic surgery or even reconstructive surgery. Now, something that you can see here in the anterior part of our thorax is that these collagen fibers runs from the shoulder area towards the middle of the body, like to the sternum in an angle. So surgeries around this area are better if they're performed following in anywhere, not only here, but if they follow this angulation. Why? Because the edges of the wound will be closer together as you cut these collagen fibers parallel. And when you have the edges of the wound closer together, repairing by the fibroblasts is much faster, and then you have less scarring. Like look here in the thigh. These lines as well tend to run in an angle from the coxal region to the patellar region. If you make a section that is perpendicular to these tension lines, you make cross section of these collagen fibers, it's hard to repair this. And then look at the edges of the wound. So these ones repair slowly and then leave more scar. Any questions so far? Okay, now how does epidermal growth and keratinization occurs? So the stem cells in your stratum basale starts dividing and differentiating. So when you have mitosis of stem cells, typically you form another stem cell and a daughter cell. The daughter cell is the one that is going to further divide and is going to differentiate into a mature cell. So as this happens, then these daughter cells in the skin starts migrating towards the stratum corneum, replacing these superficial cells that we lose. And then the keratinocytes start producing these keratohyaline proteins inside and lipids, and as they migrate up, these cells start shrinking and shrinking until they die, and then you form only an inner protein that is waterproof barrier. This is the keratin. And then our stratum corneum exfoliate more or less every 7 days, 14 days, depending, but they are continuously exfoliating. So here is this process. So you have here stem cell A. The stem cell A undergo mitosis, so this is the microtubules that I was drawing there. These are the centrioles. So then, in this process, as this stem cell subdividing into this daughter cell and this another stem cell, the other stem cell stays here in this basal region, and then this B cell, the daughter cell, moves up, and this one continues dividing and then forming more daughter cells, stem cells, but this one moves to the next layer. So from stratum basale, they will go into stratum spinosum or granulosum. Spinosum, that's the next. So then, from the stratum spinosum, this cell is going to move into the granulosum. And then if it will be thick skin, will move into the lucidum. And then from the lucidum into the corneum, that's the last. And then from there, into your mattress. That's where we leave a lot of cells. So what factors influence skin color? Well, the melanin. Not only the melanin provides the skin color, but protect us against UV exposure. So this melanin, it is produced by these melanocytes, and these melanocytes are going to secrete it to the cells that are nearby in the stratum spinosum. The stratum spinosum cells are going to grab this melanin, and that would give this skin color. We have different types of melanin depending on the amount of melanin that you have of a particular kind. It's going to be your tone of skin. Even within the classification of skin colors that we have certain tones. And as well, we have other pigments like keratin, which is found in vegetables like carrots, that can influence the skin color, as well with the oxygenation levels. If someone it looks bluish, is lacking oxygen. This is known as cyanosis. Now, there are certain diseases, poisonings by heavy metals, that also can lead to this bluish color. But as a whole, someone who has low levels of oxygen will be cyanotic. And then if someone has excess of blood, like if he's blushing, the person will have reddish color. Inflammatory reactions in your skin, erythema, it will look reddish. So certain infections can cause erythema. If you have deposits, permanent deposits of collagen, of melanin in the skin, it can be freckles, moles, or uneven melanocyte distribution, including the lack of melanocytes in certain areas of the skin. The melanocytes derive from some cells that are called neurocrest cells. And some cells don't migrate, and they leave a white spot in the skin. So here is the melanin synthesis and distribution. So this is a melanocyte. It's a large cell, and this large cell is going to extend cytoplasmic extensions towards the cells that are surrounding it. And then this melanin is going to migrate in these extensions, and then by exocytosis, this will be released, and then by endocytosis, these keratinocytes are going to pick this melanin. So, functions of a specific segments of our skin. Epidermis, it is part of the physical barriers and chemical barriers that protect us from invasion by microorganisms. So that's why we have several layers, so that the microorganisms don't penetrate that easily. And as well, we have the acid mantle, that it is produced by normal bacteria that we have in our skin, plus the sebum. So our sebaceous glands produces this oily secretion that is rich in acids that inhibits the growth of microorganisms. And then we have receptors there for sensation, that tactile cells. And then the dermis functions will be strength, elasticity, nourishment, housing of blood vessels, nerves, and glands. Subcutaneous tissue, the fat tissue that we have under dermis, insulates, form a storage of energy, and cushions and thermoregulates. Questions? Now let's look at the accessory structures of the integumentary system. So the hair has a visible portion that is called the shaft, and it is made out of keratinized stratified squamous epithelial cells. So we have only the superficial part of the integumentary system. It's dead tissue. It has a root that is embedded in the dermis, and it is surrounded by a follicle. Now, this follicle, it's an epithelial sheet that anchors this hair into the dermis, and it has this follicle, a bulb. And inside the bulb, we have a matrix which has the stem cells that helps for the hair to grow, and we have this projection within the bulb that is called the hair papilla, where we have the blood vessels that nourishes this matrix of hair cells that they're supposed to grow. Functions is to protect, like shielding the scalp, and prevent foreign particles from landing into body openings, like the eyelashes and the eyebrows. Now, the hair also acts as a sensory organ. Surrounding the bulb, in the dermis, we have the root hair plexus. It's like a nerve that wraps it around and then help to detect if there is motion within that hair. You can be blind or you can be blindfolded, and if you have earmuffs, that prevents you from hearing the wind, and if we had a window and then you basically open up the window and stick out your arm or your forearm through that window, if it was windy, you can tell the direction of the wind and as well the intensity of the wind, because that wind will move the shaft and that sends sensation into the bulb and into this hair root plexus so that you can tell what is going on. Even without doing that, if you blow air through your skin or someone blows air, you can tell that because we have these receptors. So here is the components of the hair. So this is a shaft. This is a cross section of the shaft. We have an inner part that is called the medulla, and then the cortex. So all of this is a stratified keratinized epithelium, and then this shaft is protected by a cuticle. That basically, that cuticle, it is part of this dermal root sheet, but the dermal root sheet is only found here in this region. And this is the epithelial root sheet, this is the dermal root sheet, and if you make a longitudinal section, you will be able to see the medulla, the cuticle, and then this epithelial root sheet, the dermal root sheet, and in the hair ball, which is this last portion that it is found here, you have the matrix. So this is the area where we have the cells that makes this hair to grow, and then within this part we have these capillaries that provides nourishment to these cells. Well, why in some cases you lose the hair and then you don't have as much growth? Well, it can be related to these hair papilla or to these cells. So these cells might be lost, or these capillaries, they don't provide as much nutrients. On average, we have a growth of hair of 1 millimeter per day. Okay, so I think I can stop here. Let me